Hi, and welcome back to the Sound of Science. Today, we're going to be covering one of those incredibly important topics to understand in AP biology: cell communication, how cells communicate with one another, how they signal one another, and what the reception of those signals will do. This is going to cover topics 4.1 to 4.4 in AP biology. Being multicellular, being more complex, gives organisms an opportunity to do a lot more, gives them a lot more ability, um, but only because they're able to communicate between cells. Most cells in multicellular organisms are able to communicate fairly easily because they're maintaining physical contact with each other. In fact, this is so useful. It is such a valuable tool that some unicellular organisms will also do this. Bacteria will actually live in little colonies, and it allows them to have that physical contact. The really cool thing is that those bacteria then can actually take actions all at once. We see the same thing with like slime molds in the Protist kingdom, where those slime molds will come together and form a colony when it's time for reproduction, and they send signals to one another to let each other know that they're ready, and they form a fruiting body much like a fungus would, so that reproduction can take place. Cells can send chemical signals to cells that are nearby. They don't actually have to be touching. And we're actually going to learn here in a few minutes that cells can also send signals to cells that are further away. But this direct contact, again, is so useful that cell membranes and cell walls actually have evolved modifications to allow for easier communication. Plant cells actually use this structure called plasmodesmosome, and animal cells use something called gap junctions, and it's just allows for signals to pass easily between cells. As we said, cells can communicate over short or long distances. And when it's a short distance, we're actually going to refer to that as a local signal or local signaling. And we see this if the target cell is close. Now, the receiving cell is the one that we're going to refer to as the target cell. You're going to hear me say target cell and target molecule quite a bit throughout this video, so keep those straight in your mind. Often the target cell and the signaling cell are of the same cell type in local signaling. Long distance signaling, the target cell is not local, obviously, and the signal may actually have to travel quite a distance, oftentimes through the bloodstream to get where it's going. When we see long distance signaling, usually the cell types are going to be different as well. It's two different cell types that are taking part in the communication. An example of signaling through direct contact would be, for instance, immune cells communicating with each other. So, B cells get activated when they bind to specific antigens um, with the B cell receptor. So, a virus infects a healthy cell. That healthy cell now has virus capsid stuck to the outside of it. The B cell is actually going to bind to that, and it's going to recognize that that's an invader. It doesn't belong there. This is going to activate the B cell. It's going to lead to a response that actually goes through a whole series of responses within this immune response system that ultimately is going to lead to the body fighting off the infection. Another example of using a local regulator, but not necessarily direct contact, are neurotransmitters. Neurotransmitters are nerve cells, right? And think of the cells in your brain that allow you to have thoughts, that allow you to have feelings like touch and smell and vision, and allow all those things to function. So, exocytotic vesicles are going to release certain chemicals. And these are the signaling chemicals. The signals jump across this small gap to reach the target cell. And this is actually how you are able to think. When the signals attach to the targeting cell, of course, it causes a reaction and a response. With neurotransmitters, just kind of a fun fact here, that gap that it has to jump across it's called a synapse. And the gap that it's jumping is made easier to jump by the presence of serotonin. That's why individuals that have too much serotonin inside the cell and not enough outside will oftentimes have signals in their brain that are muted. They don't feel as strongly, or they don't arrive at all. And that is what causes a lot of the feelings of clinical depression. So it is very much a medical condition, and we can see it represented in cell signaling. There are 3 steps to signal transduction, and I have them in this diagram, which is not a bad idea to draw out some of these diagrams I'm showing you. And then I have the explanation to the side. So if you start off looking at this, the signal molecule is going to be received, and it's going to bind to a receptor, which is going to be part of the cell membrane, sometimes part of the nuclear membrane, or sometimes it's something inside the cell, just depends. So it's going to bind there, and it is every bit as specific as when, like, for instance, when we talked about enzymes and how enzymes were so specific. Again, this is as specific as we saw with enzymes. Not anything can just bind to a receptor. Transduction is the part that where that signal that's being sort of relayed by the receptor is going to be converted into a form that allows the cell to have a response. And then, of course, that response is a specific cellular response to the signal molecule. It can be transcription, it can deal with production of a protein. There's all sorts of things that these signals can cause. Protein synthesis can be regulated by turning genes on or off. And one of the reactions that we see can actually be this in response to a signaling molecule. We can regulate the activity of proteins in the cytoplasm this way as well. Cascades of molecular interactions relay signals from receptors to target molecules. And these cascades, just like water cascading down a hill or across rocks or over a waterfall, it picks up speed. So that phosphorylation, those cascades, are going to oftentimes amplify the effect. So you may have a single signal molecule bind, but it could cause a large response depending on the situation. So let's talk about what really is happening here, all these steps. Signaling begins with the recognition of a chemical messenger, which we're going to call ligand, or you might hear it told as ligand, by a receptor protein in the target cell. So the ligand binding domain of the receptor is going to recognize the specific chemical messenger. Again, this is very, very specific. Now, what is the chemical messenger? It can be a peptide, it can be a small chemical, it can be a protein. There's countless molecules that it could be. The signaling cascade is then going to relay the signals from the receptors to the target molecules, oftentimes, like I said, amplifying the incoming signal. This leads to a cell response. Could be cell growth, could be secretion of molecules, or, like we mentioned earlier, gene expression. On the left of this page, I have a diagram where I've put in the steps. There's 5 steps here so that you can see them. Now, this is actually a specific response and a specific hormone, but when you take a look at it, it really just kind of gives you an overview of how this will happen. It is not a bad idea for you to resketch that and put in the numbers and what they're doing or what's happening at each step. You have hormones that are either proteins or they're steroids. Okay. Hormones are some of the most common messengers we're going to find. If it's a protein hormone, it has to have a receptor on the cell membrane. It cannot pass through the cell membrane. There has to be a receptor there that's going to allow for the transduction to occur. If it's a steroid hormone, it will actually go right through the cell membrane. it doesn't have to have a receptor there. And then ultimately, it's going to bind to a receptor on the nuclear membrane of the target cell. This is just one of those examples of when we talk about how the cell membrane functions and how it allows certain things to move through and keeps out other things. These steroid hormones are able to just zoop right through and are able to work a lot faster than the protein hormones because the protein hormones have to go through another step. So, why does all this signal matter? Well, this actually allows us to respond to our environment, not just as an individual. If you see something happen and you respond, but it actually allows you to respond on a cellular level. The environment changes, and organisms need to change in response to those changes it sees. So, for instance, what I have drawn here, and again, feel free to recopy this, this might be really helpful for you, is the epinephrine response. So, epinephrine is a molecule, it's a signaling molecule, and it's going to attach to the epinephrine receptor that's found on your cell membrane. And that is actually going to cause a an event, a transduction event that's going to lead to the breaking down of glycogen and making glucose. So, why? And again, think back. We have glycogen when we have an excess of glucose. Our body forms glycogen, it holds it in reserve until it's necessary. Well, when would you have an epinephrine signal? When you are in a situation of high stress that requires flight or fight. So the flight or fight response is actually controlled by epinephrine. Epinephrine does its thing, and then what does it do? It creates glucose from glycogen. Why would you need an influx of glucose? Well, if you're going to run or you're going to fight, you're going to need energy. And so your body works to do this on the cellular level. You prepare for your fight or flight. So let's talk gene expression and cell function and how those changes can be a response. So your pathway, your transduction pathway, can result in changes in the amount or type of a particular protein, or it can actually lead to a phenotype change because of that. Another response you might get is apoptosis. Apoptosis is programmed cell death or intentional cell death. And this is when a signal arrives to the cell for whatever reason and tells the cell essentially to ramp up production of its lysosome enzymes. The lysosomes are going to break open and the cell is going to digest itself from the inside out. This is apoptosis, and actually, this can happen in response to signals. Um, Sometimes those signals just have a way of kind of recognizing when a cell has worn out, or it needs to be replaced, or it's damaged in such a way that it could be hazardous. Let's talk bacteria. So, we said earlier that bacteria will actually live in a colony so that it can communicate with each other and respond like a group with a single response. This we actually refer to by a special term called quorum sensing. Make sure you know that. And that is our bacteria signal mechanism. It allows the bacteria to sense changes specifically in population density and to respond to their environment as a group. And you can see an example of what I'm showing you over there. But it means that bacteria, once it reaches a certain threshold of population density, can actually move to the next stage of whatever infection it has going. Changes in our signal transduction pathways can actually alter the cellular response. This is a case where a mutation matters. So mutations in that receptor protein or any component of the pathway can actually change how the signal transduction takes place. These type of changes to the structure means a change in function. Change in structure means change in function almost every single time. Anything that happens downstream in this pathway can be affected or likely will be affected. So as you can see here, we have an example of a normal pathway and a mutated pathway. And where you see the X down here in our mutant pathway, it prevents the secondary messenger system from functioning. So in this case, our chemical messenger gets received, but nothing happens. And because nothing happens, there's no response. In the normal case, the messenger gets received, and this causes the transduction pathway to be activated, and we end up with a response. Where we're going to talk about the fact that these chemicals can either activate or they can also inhibit, which I hinted at earlier. If it's an inhibited pathway, typically there's going to be some type of blocking agent that prevents the process from moving beyond a certain point. This is another one of the ways that cells can respond to signals. Either it can be a positive signal that sort of amplifies, or it can be a negative signal that stops or inhibits the response from occurring. Thanks once again for joining me here on The Sound of Science. As always, I'll see you next time.